Hello, everyone. First, I'd like to thank Nestle Nutrition Institute for organizing a very interesting and important conference. I'm excited to be part of a very excellent group of speakers and to share some research in my laboratory that is focused on understanding how diet can impact select aspects of cognitive function. For some brief background, I wanted to highlight that childhood is an important period and a period of dynamic physical as well as brain and cognitive development. Brain structure and cognitive development is comprised of several key processes which occur often simultaneously in varying trajectories until the third decade of life. This protracted development trajectory allows for health or lifestyle factors such as diet and physical activity to contribute to optimal development. Research from preclinical or animal models demonstrates that nutrition plays an important role in brain and cognition through a variety of mechanisms. For example, although the brain accounts only for about 2% of total body weight, it actually contributes to about utilization of 20% of energy. So it contributes to 20% of energy expenditure. Therefore, the brain is an organ that has a high metabolic rate and high need for nutrients. Nutrients can also play important roles in cognitive function by having a direct impact in terms of energy utilization, but also phospholipid synthesis, neurotransmitter synthesis. We also know that nutrition can impact physiological health, which may indirectly impact, or healthy physiology, which may indirectly impact cognitive health. Our laboratory has specifically focused on just a handful of these nutrients, although many nutrients are involved in brain health. Specifically, we have looked at lutein, fiber, and water. In the interest of time today, I will focus on our work in the carotenoid lutein. Carotenoids are colorful plant pigments that are found in high quantities in green leafy vegetables. They are antioxidants that provide plants protection from photooxidative stress. In humans, our bodies show preferential or selective accumulation of only a handful of carotenoids in human tissue. That could include in fat, tissue, or skin. They can also accumulate in the retina as well as the brain. Specifically, we are interested in the fact that 3 carotenoids accumulate in the macula, and these include lutein, zeaxanthin, and mesozeaxanthin, where we know these carotenoids play important roles in photoreceptor protection from photooxidation from sunlight, and they also serve as function as blue light filters. We also know that some carotenoids accumulate in brain tissue, particularly lutein appears to accumulate in brain tissue in preferential amounts. And that is very interesting because lutein, among other carotenoids, is actually one of those that isn't consumed in large quantities. It's consumed in terms of proportion of carotenoids. Lutein only contributes about 12% in our diet. However, when it comes to looking at content of the brain in terms of nutrient carotenoid accumulation, lutein accounts for 60% of carotenoid content in brain tissue. Further, what is also interesting is where lutein accumulates in the brain. This selective accumulation of lutein occurs in the brain across many cortices, and we also see this in the prefrontal cortex and the hippocampus, which is suggestive of lutein's potential role in executive functions as well as relational memory. Another important aspect of this is that because humans cannot synthesize carotenoids, their uptake is therefore dependent on dietary intake via foods and supplements, making them an attractive target for dietary manipulation. Unfortunately, our consumption of lutein and zeaxanthin is very poor. These are data from a nationally representative sample of children showing that children typically consume about 0.5 milligrams per day of lutein and zeaxanthin. However much, when we actually look at this intake relative to what would be consistent with half a cup of green leafy vegetables, for example, such as spinach, you quickly realize that we're not really getting these xanthophylls in our diet in any meaningful amounts. Therefore, low lutein intake is another hallmark of nutritionally poor diets that characterize current Western diet patterns. And our work is focused increasingly on how lutein may impact cognitive function. Another innovation in the area of lutein science has been the ability to assess lutein accumulation in the retina, which we know strongly correlates with contents in the brain. Being able to assess that non-invasively using a technique that uses macular densitometer relies on an approach that It's a biophysical approach that can quickly give us an assessment of lutein concentration or macular pigmentation, what we call macular pigment optical density in the retina. And this is about a 20-minute assessment, and we have done this technique in children and shown its reliability. And what we can do with this technique is get an idea of how well the nutritional status of somebody, particularly their lutein status in the retina, in a non-invasive manner. Interestingly, this work has been conducted among adults for several years now and has shown that several studies in adults of high quality have shown that increased consumption of lutein through diet, via foods, as well as through supplementation can reliably change or improve MPOD status, but also cognition. So these are just some papers that I'm highlighting in this area. I do want to highlight this information because as you'll see shortly, we have very limited data on the efficacy of lutein consumption and MPOD and cognitive benefits in children. However, there is compelling data in adults that makes the case for lutein consumption for macular pigmentation and cognitive benefits. In fact, there's a debate regarding whether they are at the point where we can consider dietary reference intakes for lutein. We are hoping that our work in children, some of which I'll share with you today, provides important information on this topic in children. One of the earliest studies we did in the area of lutein and cognition examined the relationship between academic achievement and lutein status, specifically looking at lutein status in the retina, which we know correlates with brain lutein. So what you're looking at here are scatter plots illustrating the relationship between academic achievement measures on the y-axis and macular pigment optical density on the x-axis among pre-adolescents between 8 to 10 years of age. And what you see is that there's a positive association. So this is a cross-sectional study across all the measures of academic achievement and macular pigmentation, even after we've adjusted for some important covariates such as IQ, socioeconomic status, age, and even aerobic fitness and sex in these participants, which is these are very interesting because they were the first indicator that the benefits that we tend to associate with lutein in adults may also be observed in children. Subsequently, we examined the relationship of macular pigmentation, optical density, and memory function, specifically hippocampal-dependent relational memory among pre-adolescent children. In this task, in this small video, you'll see participants are provided with an array of 5 sport creatures where there is a study for 10 seconds, and after this period, they are allowed a brief delay, after which the children are asked to reconstruct the spatial layout of these creatures on the screen at a self-paced stage. And during the reconstructions phase, what participants do is that they can essentially engage in a variety of different reconstruction errors. And what we can do is measure the distance and the angular differences between where a particular item is studied on the screen relative to where the participant actually reconstructs the image or the icon of the creature, and then that can give us an idea of how much error they commit in this process. If you can look at, and these data here show you the variety of different errors that can be made in terms of misplacement, the swaps that individuals could make as far as the edge resizing and also the displacement. And what we notice is that essentially we see an inverse relationship between macular pigment optical density status and relational memory, regardless of which error type you're studying across the board in these participants. Of course, also still adjusting for those important covariates I mentioned earlier. So the takeaway here is that we have also seen the relationships in academic achievement and MPOD, but we also here extend those to relational memory, which we know is dependent on the hippocampus. In addition to academic achievement and memory, we also extended the benefits of regular lutein or greater lutein status to children's attentional abilities and underlying neuroelectric function. These data illustrate a comparison between children with higher MPOD versus children with lower MPOD during a modified Erickson-Flanker task. This task assesses selective attention and has 2 conditions, one with varying degrees of attentional demands, and the incongruent condition in particular places a greater demand. What we noticed here is that we observed that the participants with a higher MPOD exhibit greater accuracy, particularly in task condition that requires greater attentional selective attention. In addition to behavioral performance, we also measured event-related potentials in these participants. Specifically, we focused on the P3, which is a component that can index attentional control and information processing speed. And this is collected using an EEG cap that's placed on the participants while they're doing the flanker task. And what we learned from these these EEG patterns was that children with greater MPOD not only exhibited greater accuracy during the task condition, necessitating greater attentional demands, they also exhibited lower P3 amplitude relative to the participants who had lower MPOD, suggesting that greater MPOD not only supports greater accuracy, but also may be related to greater neural efficiency. So these data were really interesting because they went beyond just telling us about behavioral performance, but actually perhaps giving us some indication of some benefits in the neural mechanisms that are in play when the participants engage in attentional tasks and the relationship of potential benefit of greater lutein status. So thus far, the findings that I've shared with you were cross-sectional studies. And while they're very important to highlight some important relationships, what we're really interested in is in determining, in terms of policymaking or in terms of determining causal inferences, we want to look at the experimental approaches or interventions to determine the extent to which consumption of lutein can impact cognitive function. So recently we completed a randomized control trial that provided DHA and lutein to pre-adolescents between 8 to 9 years of age. In these participants, they were asked to consume lutein DHA as part of a fortified nutritional beverage over a 9-month period on a daily basis. And what we had was a lutein dose of 1 milligram and 32 milligrams of DHA versus a non-caloric placebo. We had a lutein and DHA combination because the previous data had shown that the combination of lutein and DHA in particular is important and is efficacious in improving cognitive function. This has been work that has been done in older women in a previous trial. So as a first attempt, we utilized both DHA and lutein here. And we examined a variety of different cognitive measures, not only we looked at relational memory, attentional control. We have also collected measures on brain structure and function. And today I'll only share some of these data because we have some analyses that are still ongoing. But what was interesting, and we look at our results, was that we observed that participants in the intervention group who were receiving lutein and DHA exhibited greater benefits or improvements in their macular pigment optical density following the 9-month intervention. And these are novel data. It has not been previously shown that lutein status improves in the macula of children based on dietary consumption. So we're excited about these data. Additionally, we observed benefits for relational memory, which was assessed using a mnemonic separation task that particularly focused on pattern separation. So what we observe is that the intervention group exhibits benefits in relational memory. We all, of course, see a benefit over time in both groups, but we see that there's greater change in or improvement in the intervention group. These benefits were also observed for the attentional control findings of the study. So this was based on a modified Erickson flanker task, where what you're seeing on the right here is the comparison between the two groups, pre and post, based on their reaction time interference. And what we see is that the intervention group that receives lutein and DHA exhibits greater reduction in interference for reaction time, exhibiting greater performance following the intervention. We did not observe significant benefits for academic achievement, so it appears that these benefits of lutein and DHA in combination appeared selective for attentional control and relational memory. But as I mentioned, these data are important because they're the first demonstration that we can improve macular pigmentation following dietary consumption of lutein and DHA, and that we can also see some of these benefits for cognitive function. So in conclusion, I would like to just highlight that we know from cross-sectional studies that greater macular carotenoid status exhibits children with greater carotenoid status in the macula exhibit superior cognitive function. This is consistent with what we see in adults. Although there are a large number of trials, randomized control trials that have been conducted among adults that provide evidence of the benefits of lutein for global abilities. We have a very limited number of experimental trials in children, and I believe we have conducted only one trial in school-age children. So the early indications are that we can see these benefits even in childhood. what we're learning through these data is that lutein potentially has implications really across the lifespan. And of course, this is the very beginning of this area, and we have a lot of work to do. We need to determine, for example, what best approaches should be utilized, whether supplementation and food-based approaches are the best to use, and also measuring, really the selectivity of these benefits of lutein. And another important question is about addressing the inter-individual variability and the factors that impact lutein status in individuals, particularly in children. In conclusion, I would just like to acknowledge our collaborative team and research staff for their efforts. The work I presented today involved multiple laboratories and collaborators, including Dr. Hillman, who presented today from Northeastern University, as well as Drs. Randy and Lisa Renzi Hammond at the University of Georgia. And also I'd like to acknowledge our sponsors. Much of the work I presented today was sponsored by a project that included funding from the NIH, as well as the Center for Nutrition Learning and Memory at University of Illinois and Abbott Nutrition. So thank you again. I appreciate the opportunity to speak with you today.